Hello, everyone, and thank you for staying with us. This next panel I'm excited to introduce is going to be talking about successful importation of data into DAP, lessons learned from importing registry data into RDCA-DAP. And I'm going to kick off the panel discussion by just showing very small snippets of a really extensive report that we give back to registry submitters, so submitters of data from patient registries. And this particular example is the report that was given back to the Foundation for Prader-Willi Research, and which is also represented on this panel. It's a little tough to read, but I'm going to just highlight a couple of key points. One is that in each of the submissions of registry data that we've gotten, we have tried to, at c-path and the data team, we have tried very hard to learn about the qualities of registry data that have the most impact on a quantitative modeling effort. For example, the most regulatory impact. And with each submission, we've not only given feedback back to the contributors so that they can continuously improve the collection of data, we've also learned ourselves and added additional guidance or references to things like standardized nomenclature. I saw Medra was in our Q&A session, or in the chat, sorry, earlier. So we recommend, we have a really extensive appendix that goes with this. We recommend using, for example, WHO drug for drug names. And when that's not the case, we actually do the work, the hard work in the data management and curation team of standardizing some of the things that we can identify using standardized nomenclature. So this is just a snippet. It talks a bit about the variables that we received. It talks about missingness. I think in the next slide, if I'm not able to advance, but if someone can advance to the next slide. Oops, a little too far. There we go. Nope, still the next one. Okay, so here you see that for each of the variables, we give a little summary and we say, hey, we've gotten this. This is the percentage of time that you actually have a value within that variable. So if there are 300 patients, but only one patient had a particular value reported, then that particular variable won't be useful for scientific analysis. So I'm not going to go into any more details. I want to open it up to the whole panel at this point. I think that we have talking points, but they're all centered around this concept that submitting registry data, we at c-path would like to help guide the additional improvements in the way registry data are collected. And we also require collaborative work with patient groups and registry owners in order to learn exactly what sorts of information and guidance are needed. So with that, I want to just take a moment for each of our panelists to introduce themselves really briefly. You all know me, Amanda Borens. I lead the data science team at c-path.
I'll go. I'm Theresa Strong. I'm Director of Research Programs at the Foundation for Prader-Willi Research. I am also mom to a young man with Prader-Willi syndrome.
And I'm Will. You also know me. I'm a data engineer on the data science team here at the Critical Path Institute.
Karim? Is it possible you're muted? Karim, would you like to introduce yourself? Okay, well, while we figure out what we've got with audio, I will go ahead and jump in with a question for Will. Will, can you talk about just strategies for gathering data in such a way that it will really maximize the value of the data, especially in cases of transferring data from one place to another, one database to another?
Yeah, I think there's a couple components to that. One of the critical components, though, is ensuring that there's good metadata that's captured. And so that can come in a couple different forms. But early on, deciding what some of the maybe controlled terminology or predefined characteristics of the data you want to make sure that you capture, where we are being flexible enough that patients are able to give us their real experience, but we're not having to mine free text fields completely. While at the same time understanding what were the instructions that were given to a patient, trying to kind of annotate the data early on so that that's in both a machine and human readable format. And of course, this is easier said than done, but the more that we can try to solve that problem and work with each individual group to identify how we can help patients still enter data well, but that we're able to then interpret that down the road is one of the key components of what we're looking to see happen.
And so, Theresa, if I can jump over to you, we have a couple of talking points that we'd love to hear from you about. And one is just, what were your goals when you were designing the questionnaire that you used to collect data for Prader-Willi patients? What were you expecting to get? What were you expecting to accomplish? What sorts of outcomes are you looking for?
Well, I think our goals have evolved over time. As Dr. Neelan mentioned earlier, we have quite a few surveys within our registry. And initially, we were interested in getting a core set of data on the clinical symptoms of PWS, retrospectively collected. But as time has gone on and we've had new pharmaceutical companies come into our space, getting prospective natural history data, looking at how behavior changes over time, how environmental control influences behaviors has been really important to us, and using the registry to validate endpoints, for example. So we've had this registry now for 6 years, and it's continued to grow as we've grown. And this is, a nice natural next step for us, is to collaborate with the RDCA-DAP team, see how our data can be used in conjunction with other data sets, and also, as our data has gotten more complex, use the platform to analyze that in more sophisticated ways.
So you've made such a good point about how you've evolved over time, and I'd love it if you could give us just a few more words about exactly how you improved your registry over time. In other words, how did you hone in on more clearly accomplishing your goals with the surveys? What sort of changes did you make?
Well, they were changes in the type of data collected. For example, we are now gathering more patient experience data that would support, the impact of our disorder on the individual with Prader-Willi syndrome, as well as the entire family. And that is, supportive. That's to help the regulatory agencies really understand the unmet need in our community. And again, our prospective natural history study is gathering data on the serious medical events that occur in our population, so that we can know what the baseline of serious medical events is, so that when we're adding clinical trials on top of that, we can see if there are changes in that, and also understanding how behavior changes over time. And I think there's a lot of, obviously, over the last 6 years, there are new opportunities to get real world data that can be incorporated. And so, we look forward to continuing to expand that.
Can you hear me now, Amanda?
Yeah, Karim, please introduce yourself.
Yeah, apologies for that. So this is Karim Azer, and I'm Vice President of Systems Biology and Discovery here at Axcella Health, and really great to be a part of this panel.
Karim, I'd love to just transition over to you, building on what Theresa talked about in what are your goals as a patient group for collecting data, and how have you gone to improve that over time? She made a really important point about connecting with pharmaceutical industry partners to say, what sorts of data would you like to see? So I'd love to hear your perspective as an industry partner about what would you be looking for in a high quality registry? What would help you to say that these data are going to be useful in my discovery process?
Yeah. So from a usability or application standpoint, what I will be looking for is data, sort of multi-scale data that could speak to underlying biological mechanisms that are implicated in a disease, and as well as the translational relevance of these, if they are collected under different non-clinical conditions and non-clinical systems, if there's any data on the distributions of these biological measurements in a clinical setting in the patient population. From a data standpoint, what I'll be looking for are really four things. There's the data quality aspect, data access, data readiness, and then the analytics and the tools that allow us to be able to actually use this data in any real way. So from a data quality standpoint, it's all about the harmonization of data, any gaps or missing data elements, and that sort of framework. The access piece is ease of access, how we could actually able to get to the data, be able to the use of the data for discovery of novel potential therapies that we could advance from a industry standpoint. The other aspect is readiness, which is basically, how much data once I can ingest the data sets and registries into our platform, how much of an effort does my team have to do to get it to a point where we can actually utilize it for discovery efforts and translational efforts. And then finally, it is always helpful to have analytics tools, visualizations that are readily accessible and have been constructed by the data providers to make an initial assessment whether the data will actually be useful for the context of use. But also, as a validation point, once you ingest data, to make sure that everything went through, and as a validation step point, basically, that we can reproduce any trends that you've seen in the data from the source.
Well, that's good feedback. And I'll tell you, we haven't really touched much on the details of our curation process at c-path, but it's something that I think we're really proud of, in that we do an awful lot of work to standardize nomenclature and clean misspellings, for example, the various ways that drug names are listed. We do a lot of work to identify and account for records that we might have to exclude by going back to the original contributor. But the thing that I think is particularly salient in your point about, the quality of the data, is that it also includes things like the standardized way in which data were collected. And so I think, and I'd love to hear from everybody about this, but I think it's a particular challenge with patient registries, especially because, you have patients or caregivers who have limited time, who are doing everything that they can to expose the community to their information, their symptoms, what concerns them, what they're taking, what they're doing. And sometimes it's heartbreaking in that the data are collected in such a way that it's sort of everything that I could throw at a registry, right? And so from each of your perspectives, I'd love to hear about why that can be concerning. And I'm going to just kick it off by saying that I can remember a particular case of registry collected data where patients were uploading reports from electronic health records, but they were uploading them in a way that they were images. And so, the text being extracted from it was nearly impossible to get. And so that's one of the ways that, I think that we as an RDCA-DAP community would love to see more guidance and more official guidance, honestly, from regulatory agencies around the world about exactly how to collect registry data in such a way that it maximizes its impact. But if each of you would like to take a turn, we'll start with Theresa, please.
I think that's a great point. Our families have a lot on their plates. They're certainly, willing to contribute to the research process, but we have a very strong obligation to make sure that their data is being used to its greatest effect. So I think that's another motivation for us to be more involved in RDCA-DAP, because the feedback on making sure that we're collecting the data in the best way that we can so that it has maximum utility is really, again, an obligation to our community.
Karim, I wonder if you sort of started to put your toe in the water of talking about longitudinality, but one of the things that I've seen with registry data that can be challenging is that patients report symptoms or medications but not at regular intervals. And how does that make things challenging for you in the development process?
Right. So it's really about interpretability, and we want to make sure that we're interpreting the data appropriately, but also our interpretation then dictates how we could link these data registry with other data sets, because no data stands by itself, and typically we'll be looking at adjoining these data sets from a mechanistic standpoint with computational biology data, right? Metabolomics, proteomics, to look at underlying mechanisms, and then to build relationships, right? Are any of these markers, mechanisms, proteins correlate with the changes in symptomology that is reported in the data? So when you have quality gaps in the data, that hampers that ability to mine it and be able to interpret, to come up with understanding of, how do these symptoms emerge and so on. The flip side of it is, as an example, in a previous life, we were using registry data to build a disease progression model that was going to be used to help us quantify the pharmacological impact of a therapy that we're working on. And that actually worked really well, but that was, it worked well because there was significant investment from the community, as well as, honestly, on the industry side, willingness to put in the effort to around data quality and access, as well as working with the patient groups to make that happen and carry forward and consolidate all this data into a single platform. So there's tremendous value in what you're proposing here and what you're doing, and we're very excited from an industry standpoint in being able to leverage that, on our front for discovery and translation into the clinic.
Well, I would give you the opportunity to close out this topic and talk to us a little bit about the qualities of what you would consider high quality registry data, right? And maybe just a little bit about your experience as part of the data science team over the last year in bringing in, ingesting some of the registry data, and then adding appendices and guidance in these reports. Can you kind of summarize some of the highlights of what it is that we're able to salvage after the fact versus what patient groups can do to prevent mistakes or prevent problems with the data before building the registry?
Yeah. So I think that's a great point. I think both Karim and Theresa kind of got at this with the context of use and making sure that we're matching our endpoints and our outcomes against something that is actionable, and ensuring early on that there's a good design that is enforcing sort of that relationship between the biologics and between the lived experience of patients. And so, I think that's kind of a lofty goal, but keeping that in mind, once we have that context, there's some things that we could do technically to help make sure that those registry data are collected well. So beyond just good metadata, enforcing things like saying there has to be a date value, and that this date value can't be, later than today, and it can be in the future. That's something that can go a long way, and it's a pretty easy, thing to do. But it's that experience of once you've been through this a couple times, you're like, okay, well, clearly people didn't have this experience in 2050. This was a typo, and they're entering data. And so the more that we can do to help expedite some of these technological components so that we ensure that there's good quality data collected at the start will translate in the long run to really getting that quality of use and helping us make sure that what we're looking to accomplish, we're able to get to that endpoint quicker.
I'll just wrap it up then by saying thanks to Theresa, thanks Karim, thanks to Will for all of your comments, and also keep your eyes peeled. This fall, we're planning to have submit a publication that is around best practices for collecting registry data for maximum regulatory impact. And with that, I'll pass the baton to the next speakers.